We'll pick up where we left off last time, and that is with the components of the sarcomere. We said that the Z discs were the end points, the boundaries, if you will, of the sarcomere. The A band or the anisotropic band will be the darker layers, darker sections of our sarcomere. We have to remember that when we described our skeletal muscle cell, we said that they were, I talk about them as being a striped worm. We have alternating bands of light and dark. The A band is going to be that dark band, because remember my goofy trick, dark, right? A, dark, right? In the center of the A band, we have something called the H zone. And although we have our thick and thin filaments overlapping, in the H zone, it's an area where it's only made up of the thick myofilaments. The thin myofilaments aren't nearly as long, which you'll see in a moment when we get a diagram up for you. Next, we have the I band or the isotropic band. So again, this makes sense. If the anisotropic band is the darker area, the I band or the isotropic band will be the lighter band. And again, light, there's an I in light for isotropic. It is composed of only the thin myofilaments, so that makes sense. The thin myofilaments are not going to be as thick and as dark as either just the thick alone as in the H zone or the doubling up of both types of myofilaments. Hopefully this will help. What we see here is that same diagram of a sarcomere, and we have our Z discs. We have our A band kind of in the center, and right in the center of that is the H zone. Then we have our I bands to the sides. Above that, we have an image from an electron microscope that's showing us really the almost to the molecular level what we're looking at. And they're superimposed on each other. So at the edges, those really dark lines, dark vertical lines, are the Z discs. And again, if you look below, they're right in line with our diagram. Then, when we look at the sarcomere, you see that we alternate our thin and our thick myofilaments, right? That they're both going to be anchored into the Z disc, but they alternate. And you see that that A band is where the thick and the thin myofilaments overlap one another, where we're going to have that area where both thick and thin are present. To the sides of that, we see a lighter color, right? And that's where we see only the thin myofilaments. That would be the I band. So this is the setup of our sarcomere, and this setup is what's going to allow any one of your skeletal muscles and your cardiac muscle, to be perfectly honest with you, but jumping ahead, it's going to allow those cells to contract and allow your muscles to move the way that we know them to do, right, just from everyday life. This is a kind of a nifty diagram. This is showing you the interplay, the interconnection between the thick and the thin myofilaments. You will see that our golf club heads of the myosin are quite evident. If you're looking at your myosin filament, you see there's kind of a squiggly bit, right, coming back from the head. That is the hinged area that we spoke about. Above that, you see our actin molecules, our troponin and our tropomyosin, and they're in very close proximity because our myosin head will connect with the active sites of actin when they are uncovered. And from this diagram, it looks to me that they are in fact uncovered. It's tough to say, but that would be my guess. Now, muscle contraction. We refer to how our muscles contract, our skeletal muscle cells, how they contract. We use the sliding filament theory of contraction. This was identified by Hugh Huxley way back in 1954. And yes, it is older than I am. Thanks for asking, smart Alex. But this is what we know to be the mechanism of contraction. Many of you are sitting there thinking, well, gosh, there's got to be something new or better. We must have come up with something more innovative or precise in all of those years since 1954. The fact of the matter is, Hugh Huxley hit a home run on this one. Way back then, he had it nailed, right? Exactly how he has described it is today what we know how it happens, right? We've looked at it further. We've gotten more precise in our ability to investigate, and it still keeps coming back to this sliding filament theory. Okay, so again, if it's not broke, don't fix it. Regulation of contraction. What causes a contraction to happen? What causes a contraction to stop happening? As we talked about at the beginning, right? Nerve impulse leads to the contraction. That nerve impulse is going to cause an increase in the calcium ion concentration within the muscle cell. So now remember, at this point, we have that specialized endoplasmic reticulum, that sarcoplasmic reticulum, and its job is to store away, to hold on to calcium ions until they're needed. Well, apparently during a contraction is when they're needed. So they're going to leave the sarcoplasmic reticulum and kind of float around and move around all of those protein filaments that we've been talking about. So let's look a little closer at the sliding filament mechanism. During a contraction, the thick and the thin myofilaments slide past one another. Keep in mind, right, and this might be a good idea, have your textbook open with the diagram of a sarcomere, so as we're talking about this sliding filament mechanism, you can look at that, that diagram and merge the two, merge the words with the picture, and hopefully that will help you flesh this all out. So anyway, during a contraction, the thick and the thin myofilaments slide past one another. Remember, both of those myofilaments are anchored in the Z disc. The thin myofilaments move towards the center of the sarcomere because they dead end, they attach right to the Z disc. The thick myofilaments are kind of suspended in the middle of the sarcomere. So as they move, as they slide towards each other, the thin myofilaments are going to move towards the center. I wish you could see me. My hands are sliding, my fingers are sliding towards one another. The myofilaments don't change length. Okay, the myofilaments are as long as they are long. The sarcomere is going to shorten. Okay, think about that for one sec, right? The myofilaments aren't going to change length. The protein is as long as the protein is going to be. However, the sarcomere is going to shorten. Well, how is that going to work? Well, if you think about it, if those thin myofilaments are moving towards the center and they're anchored onto the Z disc, as they move towards the center, they're going to drag that those Z discs, with them and shorten, cause that sarcomere to get shorter in length. This sliding causes the sarcomere to shorten because the Z discs are going to be drawn towards one another. This mechanism can only occur if there's plenty of calcium ions and energy in the form of ATP that's available. Now, if you take a moment and think back about things that we've really stressed since we've been talking about the muscle system, it's this presence of calcium ions, right? We have a specialized organelle that is tasked with simply storing calcium ions. The cytoplasm of a skeletal muscle cell is completely different in order to be able to generate lots and lots of ATP, right? We have myoglobin that's going to help hold on to oxygen. We have glycogen, which is going to store glucose. We have more than the average number of mitochondria. All of these things are coming into play in order for this sliding mechanism to work. Here's a diagram that you can probably get from your textbook. This one isn't from your textbook. Now, if you look up in the upper left-hand corner, I call that the gecko toe. If you think about what a gecko toe looks like, right? And again, Google it. It's going to be kind of a flattened disc. That is the end point of our neuron. We said just a slide previous that we need we need a neural impulse. Well, that's going to come from a neuron, and that's going to travel along that red line, which is representing the sarcolemma, and it's going to allow it to drop down through it looks like a yellow chute. You'll remember that we had yellow tubes. What did we say they were called? Right, those transverse or T tubules. That allows the nerve impulse to get down deep in, and that's what we're seeing happening. What looks like maybe udders on a cow sideways, maybe? That is the sarcoplasmic reticulum. And even if I didn't know it by how it looks, it's filled with calcium ions, and that's the job of our sarcoplasmic reticulum. Okay, so these are the players that we're looking at. So, just prior to a contraction, the amount of calcium ions in the sarcoplasm is quite low. That's because the sarcoplasmic reticulum actively pumps calcium ions and stores those calcium ions in the sarcoplasmic reticulum. You'll remember when we talked about active transport, when we talked about our sodium-potassium pump, we said that the reason was there was a physiological reason to keep the sodium concentration high on one side of the membrane and the potassium concentration high on the other side of the membrane. Here's another example of that. In order for skeletal muscles to contract the way we know that they do, we have to have a very, very low concentration of calcium ions in the sarcoplasm, which means we're going to drag those calcium ions kicking and screaming once they diffuse, because they're going to diffuse, because we know diffusion happens, right? I've got the t-shirt to prove it. Once diffusion occurs, we're going to drag those calcium ions back into the sarcoplasmic reticulum to keep that concentration gradient in place. As a nerve impulse moves along the sarcolemma and into the T-tubule system, like we just saw in that diagram, it causes the calcium ion release channels in the SR to open. SR is the abbreviation for sarcoplasmic reticulum. So as this nerve impulse goes along the sarcolemma and drops down through our transverse tubules, that is the signal for the release channels to open, for the doors to open for the calcium ions. So what are the calcium ions going to do? Right, they're super overcrowded. They're jam-packed into the sarcoplasmic reticulum. We finally open the doors. What are they going to do? They're going to diffuse like crazy, right? They are going to flood into the sarcoplasm, and the only place to go is around those myofibrils, our thick and our thin myofilaments. So they're going to move as best they can all around those myofibrils. And let's think for a moment before I change the slide. Who do we know really, really likes to hang out with calcium ions? Was it the actin, the myosin, the tropomyosin, or the troponin? It was the troponin. So it makes sense that these calcium ions are going to go and find their friend. They're going to go and be attracted to the troponin. Here's our same diagram, okay? The little teal dots are representing what we call neurotransmitters. They come from the neuron, and they carry the message over to the sarcolemma, to the part of the muscle cell. The arrows are showing us that that impulse is going to travel along the sarcolemma, go down through our transverse tubule, and once that goes down through, those arrows are representing the impulse going down, it causes the calcium ions to escape, to get out of the sarcoplasmic reticulum, okay? So that's going to allow our next step, okay, to happen. Because the sliding mechanism theory just kind of says, okay, these proteins are going to slide past each other. We know that it happens once the calcium ions are released, but what's going to allow that to happen? This brings us to the ratchet theory of contraction. I know some of you are giggling because it says ratchet. There was a long time I didn't understand why folks laughed at that name. What I'm going to ask you to do, right, I'm going to end a little early for this lecture, is to take some time, get your textbook out, and read through the ratchet theory of contraction before you listen to our next lecture, so that you're going to have heard all of these words before we start talking about them. If we were face to face, I would assign you these pages to read before we talk about it, so that probably reading it, it's not going to make a whole lot of sense to you. But now, once you listen to the lecture, it will become much more clear for you.